Welcome to Unit 5.3 on the cells covering reproduction and meiosis. I'm Wendy Nofke and I'm going to present this video clip for you today. What we've looked at up till this point has been asexual reproduction. This just involves a single parent cell that creates offspring that are genetically identical. The advantage to this is you can increase the number of cells very rapidly. It's a very fast form of reproduction. Some examples include binary fission, where the parent cell splits into two identical daughter cells, fragmentation, where the parent cell breaks into pieces and each of the pieces forms a new organism, and budding, where a bubble-like bud forms on the parent cell and that breaks away to make a new organism. With sexual reproduction, this is going to involve two parents. The parents are going to form offspring, or form gametes, which are the reproductive cells which will unite to form the offspring. These gametes are haploid in that they contain half the number of chromosomes that the parent cell has. This is important because when they form a zygote, you'll have the fusion of the two gametes in a process called fertilization, and you'll end up with the full number of chromosomes again. A diploid organism has 2 times the number of chromosomes as the gamete. So during meiosis, the number of chromosomes is reduced by half. There are 2 divisions that go on, meiosis 1 and meiosis 2. Each has 4 phases. During the 2 main phases, meiosis 1 and meiosis 2, each are going to have the same 4 phases you see in mitosis: the prophase, metaphase, anaphase, and telophase. In meiosis 1, the homologous chromosomes will separate into daughter cells. In meiosis 2, there is no copying of the genetic information. The chromosomes divide, and each daughter nuclei has half the genetic information with a single copy of the chromosome. So if we look at meiosis 1 in a little bit more detail, we've got prophase 1. Here you're going to have the nuclear envelope break down and disappear. You're going to form the chromosomes and the mitotic spindle, and the homologous chromosomes are going to pair. This is unique to prophase 1, having these homologous chromosomes. In metaphase 1, the paired homologous chromosomes are going to align along the metaphase plate, or the mitotic spindle in the center. During anaphase 1, the homologous chromosomes will pull apart and separate towards the opposite poles. So looking under the microscope, this would look very similar to mitosis at this point, except you have homologous chromosomes instead of just single chromosomes. In telophase 1 in cytokinesis, the spindle breaks down, you form a new nuclear membrane. The 2 daughter haploid cells are going to split apart with a random assortment of chromosomes from the homologous pairs, and the rest of the cytoplasm divides. In meiosis 2, we have those same 4 phases again. In prophase 2, the nuclear envelope breaks down and the mitotic spindle and the centrioles separate. In metaphase 2, the sister chromatids are going to align. So you had the homologous chromosomes align in meiosis 1, the sister chromatids align during meiosis 2. In anaphase 2, the sister chromatids will separate. During telophase 2 in cytokinesis, you'll have the spindle break down, the new nuclear membrane forming, and now you'll have 4 haploid cells. Each are going to have a unique combination of chromosomes formed as a combination from what the parent cell had. So this process of developing or forming these haploid cells is called gametogenesis. The gametes are your reproductive cells, the sperm and the egg. It is slightly different in humans for males and females. For the female, you start out with your 1 cell. It undergoes meiosis 1 to make 2 cells. One of those cells will end up becoming a polar body, and then you'll undergo meiosis 2 and you'll make the mature ovum, and the remaining 3 cells become polar bodies. So you end up with a total of 4 polar bodies that end up being formed from the female. In the male, you have your sperm production, and you start out with 1 cell. It undergoes meiosis 1 to form 2 cells. Those will undergo meiosis 2 to form 4 cells. one of the advantages of sexual reproduction is the genetic variation that it allows. It allows you to form offspring that are genetically unique, so it's going to introduce more varieties of genes into the population. Some ways that this can happen: you can have crossing over, which is going to be exchange of the genetic material between homologous chromosomes. So when you have two homologous chromosomes, if they want to, they can exchange a couple of the genes on that chromosome with its homologous pair. With independent assortment, the homologous chromosomes are going to be randomly distributed into the daughter cells. So when there are two homologous chromosomes formed, say for example you have A and B, one daughter cell may get A from the first chromosome, A from the second chromosome, B from the third chromosome. The other daughter cell will then get B from the first chromosome, B from the first chromosome, and A from the third chromosome. So this allows you to create gametes that are unique. When the two gametes unite to produce new offspring, you get even more variation in there because you've had the other parent's chromosomes undergo crossing over and independent assortment as well. There are a variety of sexual reproduction lifestyles. They fall into three main categories. The haploid life cycle is going to be seen in many single-celled organisms. These organisms spend the majority of their life as haploid gametes. So that would be like saying you spent the majority of your life being only the egg or only the sperm. With the diploid life cycle, the majority of the organism's life cycle is the diploid adult stage. It will produce haploid gametes that fuse to form zygotes. This occurs in animals, so you only spend a short time being the two separate sperm and egg. You spend the majority of your life as a diploid individual. Alternation of generations is another variety where you alternate between being in a haploid and a diploid stage. This is seen in plants, algae, and some protists. So you may have one form of the plant that you'll see when it's in its haploid state or gametophyte state, and then you may have another form of the plant that you'll see when it's in its diploid or sporophyte phase. These two forms of the plant can actually appear significantly different in some types of plants.